Before there was language, there was chemistry. Right now, beneath your skin, a conversation is unfolding, not in sounds or signals you could hear, but in molecules, invisible messengers drifting between cells, binding to receptors, triggering cascades that ripple through tissues. Every heartbeat, every breath, every thought you're having depends on cells asking questions and receiving answers in a chemical dialect perfected over billions of years. But here's what's extraordinary. These aren't random exchanges. They're coordinated, precise. A single cell in your developing embryo receives dozens of chemical signals simultaneously, integrates them like a biological computer, and makes decisions, whether to become a neuron or a muscle cell, whether to divide or stay still, whether to live or die for the sake of form. Your body wasn't built by a blueprint, it was negotiated. 37 trillion cells, each one listening, responding, coordinating with neighbours through an ancient molecular language that transforms a single fertilised egg into eyes that see, hands that create, a brain capable of wondering how it all works. Tonight, we're going to decode that language, and what we discover might change how you understand not just biology, but communication, intelligence, and the emergence of order from chaos itself. This is Omni, stories about life, matter, and the mystery in between. Like the video, drop your thoughts in the comments, and subscribe if you believe curiosity should never end. Picture a single cell in an embryo 7 days old. It sits wedged between dozens of identical neighbours, each one a perfect copy of the others. Same DNA, same proteins, same potential to become anything. A heart cell, a nerve cell, skin, bone, blood. But somehow, this cell needs to know where it is. Not in space exactly, in pattern, in the architecture of a body that doesn't exist yet. How does it know? The answer comes not from within, but from without. From its neighbours. They're speaking to it constantly in gradients of molecules that form invisible slopes across the developing tissue. High concentration here, low concentration there. The cell reads these gradients the way you might read the temperature change walking from a warm room into cold air. It's positional information, encoded chemically. Scientists call these signalling molecules morphagens. The word means shape maker. And that's exactly what they do. In the 1950s, mathematician Alan Turing proposed that patterns in nature, stripes on zebras, spots on leopards, the arrangement of fingers on your hand, emerge from chemical reactions. Two substances diffusing at different rates, activating and inhibiting each other. From that simple dance, complexity blooms. But Turing was theorizing. It took decades before biologists found the actual molecules doing this work. By the early 2000s, researchers studying fruit fly and zebrafish embryos had mapped some of these morphogen gradients in exquisite detail. They could watch, in real time, as a protein called sonic hedgehog spread from cells along one side of a developing spinal cord, creating a gradient that told cells their position along the body's midline. Cells exposed to high concentrations became one cell type. Lower concentrations triggered different fates, threshold after threshold, dividing the embryo into precise developmental territories. Suggested footage: zebrafish embryo time-lapse showing fluorescently tagged morphogens spreading across transparent tissue, rainbow colour-coded to show concentration. But morphogens are just one language in a larger vocabulary. Cells communicate through at least 4 distinct molecular systems, each with its own grammar and purpose. There are morphogens that form gradients. There are growth factors that tell cells when to divide. There are cytokines that coordinate immune responses. And there are direct cell-to-cell channels called gap junctions, microscopic tunnels that physically connect the interiors of neighbouring cells, allowing them to share small molecules instantly. According to research published in 2024 by the journal Cell Communication and Signaling, these communication pathways don't operate in isolation. They crosstalk. A signal received through one pathway can modulate how a cell responds to signals from another. The result is a computational network of staggering sophistication, billions of messages processed every second, integrated into coherent decisions that build and maintain your body. Consider what happens when your heart beats. Each contraction requires the coordinated firing of millions of cardiac muscle cells. They don't have a central controller telling them when to contract. Instead, they're linked by gap junctions composed of proteins called connexins. When one cell depolarizes, its electrical state shifts. Ions flow through these junctions into neighboring cells, triggering their depolarization. The signal propagates like a wave, cell to cell to cell, fast enough that your entire heart contracts as one synchronized unit. Studies from early embryonic development reveal that gap junctions appear remarkably early. During compaction, when an 8-cell embryo's cells press together and begin coordinating for the first time, connexin 43, one of the proteins forming these junctions, starts appearing on cell membranes precisely when cells need to start making collective decisions about which will become the inner cell mass and which will become the outer trophoblast layer. Without these conversations, development fails. Knock out the genes for key morphogens in experimental animals, and embryos develop with missing structures, misplaced organs, bodies that can't tell front from back or left from right. Disrupt gap junctions in the heart, and you get arrhythmias, chaotic, uncoordinated beating that can be fatal. Interfere with growth factor signaling, and cells divide when they shouldn't, leading to tumours and cancer. But when the conversations work, when cells listen, integrate, respond, the result is breathtaking. From a single cell, differentiation cascades outward. A gradient of one morphogen triggers the expression of genes that produce other morphogens, creating new gradients at right angles to the first. These intersecting slopes divide the embryo into quadrants, then eighths, then increasingly fine subdivisions. Like a sculptor working from a rough block to fine detail, chemical conversations refine the body plan with precision that approaches the limits of what molecular noise should allow. Research from Nature Communications in 2022 revealed just how precise this system can be. In the developing neural tube of mouse embryos, morphogen gradients specify the positions of different neuron types with an accuracy of about 5% of a cell diameter. That's remarkable, given that individual morphogen molecules are subject to random diffusion, degradation, and molecular noise. The embryo uses multiple strategies to achieve this precision. It reads gradients over time rather than at a single instant, allowing cells to average out random fluctuations. It uses opposing gradients from both ends of the tissue, letting cells triangulate their position. And it employs feedback loops. Cells that receive signals produce their own signals that reinforce and refine the original pattern. The system is robust. You can remove half the cells from an early embryo in many species, and the remaining cells will reorganize, rescale their signaling, and produce a normal, if smaller, individual. The gradients adapt, the thresholds adjust, the conversation continues. But the most profound question isn't how cells communicate, it's what they're computing. When a cell receives a dozen different signals simultaneously, morphogens telling it where it is, growth factors telling it whether neighbours are crowded, mechanical forces from surrounding tissue, metabolic signals about nutrient availability, it doesn't just respond to each independently, it integrates them. Inside the cell, signalling pathways converge on gene regulatory networks, complex circuits of transcription factors that activate and repress each other in patterns that depend on the combination of inputs. A 2024 study on cell signaling dynamics revealed that these networks don't produce static outputs. They're dynamic. The same combination of inputs can produce different responses depending on the order signals arrive, their duration, their amplitude. Cells are reading not just what signals they receive, but when and how, extracting temporal information from chemical gradients. It's computation without consciousness, decision-making without deliberation. A biological intelligence that emerged not by design, but through 4 billion years of evolutionary selection for systems that could reliably build complex, viable organisms from simple starting points. Tonight, we're going deep into this molecular conversation. We'll trace how the first chemical signals likely emerged in early multicellular life. We'll explore the specific molecular mechanisms, how receptors catch signals, how signaling cascades amplify weak signals into strong responses, how cells remember what they've been told even after the signal fades. We'll discover how errors in these conversations lead to birth defects and disease, and we'll end by asking what this reveals about the nature of biological information itself. Because understanding how cells talk to each other isn't just developmental biology. It's the foundation of regenerative medicine, cancer therapy, and perhaps our best window into how life organises itself from molecular chaos into the staggering coherence of a living body. Let's begin where all complex life begins, with the decision to stop being alone. The first cells to speak to each other probably didn't mean to. About 800 million years ago, somewhere in the shallow seas of a planet still alien to anything we'd recognise as complex life, single-celled organisms began experimenting with staying together after division. Not always, not permanently, but sometimes, under certain conditions, daughter cells didn't separate. They formed clusters, clumps, colonies held together by sticky proteins on their surfaces. This wasn't cooperation, not yet. It was accident becoming experiment, repeated enough times across enough generations that natural selection could grab hold of it and start to sculpt. But here's the problem colonies immediately faced: conflict. When genetically identical cells cluster together, they're sharing resources. Nutrients diffusing through water reach the outer cells first. Light, if they're photosynthetic, hits the surface before penetrating deeper. And if these cells can still divide independently, there's an evolutionary temptation for any one cell to defect, to take more than its share, to reproduce faster than its siblings, to cheat the collective for individual gain. According to research published in the American Journal of Physiology in 2013, cell signaling likely evolved as a mechanism of conflict mediation. Before multicellular organisms could build bodies, they needed ways to keep their component cells aligned toward shared evolutionary interests. Signaling molecules became the language of negotiation, the chemical contracts that said, if you detect this molecule, respond this way. If you produce this molecule, your neighbours will respond that way. The earliest signals were probably metabolic byproducts, simple molecules like cyclic AMP, a derivative of ATP that cells already used internally to regulate energy metabolism. But when cells started staying close together, these internal signals became external cues. A cell experiencing nutrient stress might increase its cyclic AMP production. Neighbouring cells, detecting elevated cyclic AMP in the extracellular space, could interpret this as a warning: resources are scarce, adjust accordingly. Modern slime moulds still use this system. When individual amoeba-like cells exhaust their food supply, they begin secreting cyclic AMP in pulses. Other cells, detecting these pulses, move toward the source and begin secreting their own cyclic AMP, creating a wave that propagates outward, recruiting more cells. Tens of thousands aggregate into a slug-like multicellular structure that migrates toward light, then transforms into a fruiting body, some cells sacrificing themselves to form a stalk, while others become spores that will disperse and start the cycle again. It's cooperation, emerging from chemistry. No cell understands the larger pattern. No cell chooses to sacrifice, but the signalling system coordinates individual responses into collective behaviour that enhances survival when times are hard and dispersal when resources return. The transition to obligate multicellularity, organisms that are always multicellular, that die if you separate their cells, required more sophisticated signalling. Not just warnings about stress, but constant conversations about identity and position and function. Research from 2024 analysing the genomes of filamentous algae, that are the closest relatives of land plants, reveals that the transition to true multicellularity involved an explosive expansion of genes encoding signalling molecules. These organisms don't just have a few signal types, they have dozens of different receptor families, hundreds of downstream signalling proteins, elaborate networks that allow cells to integrate multiple inputs simultaneously. Why so many signals? Because multicellular life faces a fundamentally harder problem than unicellular life. A single-celled organism responds to its environment directly. Food nearby? Move toward it. Temperature too hot? Adjust your membrane composition. The cell is the decision maker and the executor, the sensor and the actor, all in one package. But in a multicellular organism, cells are insulated from the environment by other cells. A cell deep in a tissue doesn't directly experience what's happening outside. It needs to be told. It needs information relayed from the cells, at the surface, interpreted and passed along through layers until the message reaches the cells that can do something about it, and cells need to coordinate. When you decide to stand up, billions of muscle cells must contract in precise sequence, triggered by signals from neurons, which themselves were triggered by decisions made in your brain by other neurons integrating sensory information about balance and intention and where your limbs are in space. No single cell knows you're standing up. Each knows only its local instruction. Contract now or stay relaxed, or adjust your contraction strength based on resistance. The solution evolution found was layered communication networks. At the most basic level, cells needed to identify themselves and their neighbours. Friend or foe, same tissue type or different, self or pathogen. This required cell surface proteins that could recognise and bind to complementary proteins on other cells, adhesion molecules that don't just stick cells together, but also trigger signalling cascades that say, I'm touching someone like me. Adjust gene expression accordingly. Then cells needed to know where they were in the developing organism. This is where morphogens entered the picture, signalling molecules that don't just turn on or off but form gradients. High concentration here, low concentration there, creating a spatial map that cells could read chemically. The word morphogen comes from the Greek, morphe, meaning form, and genes, meaning producing. Form-producing molecules, chemicals that literally build shape by telling cells what to become based on where they are. British developmental biologist Lewis Wolpert formalised this idea in 1969 with what he called the French flag model. Imagine you're trying to create a French flag, blue, white, red stripes, using cells that are initially all identical. How would you do it? Wolpert proposed that a source of morphogen at one end of the tissue creates a concentration gradient. Cells measure the morphogen concentration they're experiencing and compare it to threshold values. High concentration, above threshold 1, become blue. Medium concentration, between threshold 1 and 2, become white. Low concentration, below threshold 2, become red. The beauty of this system is its simplicity and scalability. The same gradient that works in a small embryo can work in a larger one, as long as the thresholds scale proportionally. The same molecular logic that specifies 3 cell types can specify 30 by adding more morphogens, more thresholds, intersecting gradients that divide space into finer and finer regions. But Wolpert was working theoretically. He proposed the mechanism without knowing which actual molecules did this work. The breakthrough came in the 1980s and 1990s with fruit flies, Drosophila melanogaster, those tiny insects that swarm overripe bananas and happen to be one of the most intensely studied organisms in biology. Developmental biologist Christian Nusslein-Volhard and geneticist Eric Wieschaus systematically mutated fruit fly genes and watched what happened to embryo development. Some mutations produced flies with no segments, others produced flies with segments in the wrong order, or reverse polarity, or duplicated structures where there should be one. By mapping these mutations, they identified the genes and their protein products that create the body plan. Many turned out to be exactly the kind of morphogens Wolpert had predicted. Proteins like bicoid, which forms a gradient from the front to the back of the embryo and tells cells whether they're in the head region or the tail. Proteins like decapentaplegic, mercifully shortened to DPP, which forms gradients that specify dorsal versus ventral, back versus belly. These weren't abstract theoretical constructs anymore. They were real molecules you could tag with fluorescent markers and watch spreading through living embryos, concentrations highest at the source and declining with distance, cells responding with different gene expression patterns depending on how much morphogene they encountered. Nusslein-Volhard and Wieschaus won the Nobel Prize in 1995. Their work revealed that development isn't guided by a blueprint encoded somewhere and followed passively. It's negotiated, cell by cell, through chemical conversations that unfold in real time, responding to signals, making decisions, building a body through distributed computation. But identifying morphogens raised new questions. How do cells actually detect these molecules? What happens at the molecular level when a signal arrives at a cell surface? The answer begins with proteins that span the cell membrane, bridging the outside world and the cellular interior. These proteins are called receptors, and understanding how they work means understanding how cells translate chemistry into action. A receptor is a lock waiting for a key. More precisely, it's a protein that spans the cell membrane, one end floating in the extracellular space, the other dangling in the cytoplasm, shaped specifically to recognize and bind to certain molecules and nothing else. When the right molecule arrives, the receptor changes shape, and that shape change is a signal, translated into action. The specificity is astonishing. Your body contains tens of thousands of different proteins. The extracellular fluid is a soup of molecules, nutrients, waste products, ions, hormones, growth factors, immune signals. Yet a given receptor will bind only to its designated ligand, ignoring everything else, distinguishing between molecules that differ by just a few atoms. This molecular recognition works through complementary shapes and chemical properties. The binding site on the receptor has a particular three-dimensional contour, pockets and ridges, charged regions and hydrophobic patches that matches the shape and chemistry of its ligand like a hand fitting into a glove. When the ligand approaches, weak chemical forces, hydrogen bonds, electrostatic interactions, van der Waals forces, pull it into place. The two molecules click together, and then something remarkable happens inside the cell. The most important class of receptors for growth and development are called receptor tyrosine kinases, or RTKs. According to research published in Signal Transduction and Targeted Therapy in 2024, humans have 58 different RTKs organized into 20 families. These receptors control cell growth, differentiation, survival, migration, essentially everything a developing organism needs to coordinate. An RTK has three parts. The extracellular domain contains the ligand binding site. A single alpha helix spans the membrane, anchoring the receptor. And the intracellular domain, the part hanging in the cytoplasm, contains a tyrosine kinase, an enzyme that can attach phosphate groups to tyrosine amino acids on other proteins. When no ligand is bound, the receptor floats in the membrane, usually alone, inactive. But when a growth factor, say epidermal growth factor or EGF, binds to the extracellular domain, it doesn't just stick there. It changes the receptor's shape in a way that causes two receptors to come together, forming a dimer. This is the critical step. Two receptors held side by the bridge of ligand binding between them. Once dimerized, the intracellular kinase domains activate. Each receptor phosphorylates the other, adding phosphate groups to specific tyrosine residues on its partner. This autophosphorylation creates docking sites for other proteins, recruitment platforms where the next stage of the signaling cascade can assemble. The discovery of this mechanism came in pieces across decades. In the 1960s, Stanley Cohen identified epidermal growth factor, a small protein that stimulates cell division in skin cells. In the 1970s, researchers discovered that when EGF binds to cells, certain proteins become phosphorylated on tyrosine residues, an unusual modification that seemed connected to growth signaling. By the 1980s, it became clear that the EGF receptor itself was a kinase, that it phosphorylated itself upon activation, and that this autophosphorylation was essential for downstream signaling. But the exact structural mechanism, how ligand binding leads to kinase activation, remained mysterious until 2002, when crystallography studies revealed the asymmetric dimer structure. The two kinases in the activated receptor don't just sit side by side. One kinase domain acts as an activator, touching the other kinase domain in a specific way that unlocks its enzymatic activity. It's an allosteric mechanism, activation at a distance through shape change, reminiscent of how cyclin activates cyclin-dependent kinases to drive the cell cycle. Once activated, the phosphorylated tyrosines on the receptor serve as binding sites for adapter proteins containing specialized recognition domains. The most common are SH2 domains, SRC homology 2 domains, protein modules of about 100 amino acids that specifically recognize phosphorylated tyrosine residues in particular sequence contexts. A protein called GRB2, short for growth factor receptor-bound protein 2, contains an SH2 domain that binds to phosphorylated RTKs. When GRB2 docks on an activated receptor, it brings along other proteins through two additional domains called SH3 domains. One of these recruited proteins is SOS, son of sevenless, named after the Drosophila receptor where it was discovered, which activates a small protein called RAS, and this is where the signal amplifies. RAS is a molecular switch. In its inactive state, it's bound to GDP, guanosine diphosphate. When SRS interacts with RAS, it causes RAS to release GDP and grab GTP, guanosine triphosphate, instead. GTP-bound RAS is active, and active RAS can now recruit and activate a protein kinase called RAF. RAF activates another kinase called MEK. MEK activates another kinase called ERK. This cascade, RAF to MEK to ERK, is called the MAP kinase pathway, short for mitogen-activated protein kinase. It's one of the most fundamental signaling pathways in eukaryotic cells, present from yeast to humans, used in contexts ranging from development to immune response to memory formation in neurons. Each step in this cascade amplifies the signal. One activated receptor might activate dozens of RAS molecules. Each RAS might activate multiple RAF molecules. Each RAF activates multiple MEK molecules. By the time the signal reaches ERK, the last kinase in the chain, hundreds or thousands of ERK molecules might be activated from a single receptor binding event. Activated ERK phosphorylates, transcription factors, proteins that enter the nucleus and bind to DNA, turning genes on or off. The signal that started as a single growth factor molecule binding to a receptor on the cell surface has now altered which genes are being expressed. The cell might start producing proteins needed for division, or differentiation, or migration. The chemical message has been translated into biological action. But this is just one pathway. RTKs can trigger multiple signaling cascades simultaneously. Some RTKs activate PI3K, phosphoinositide 3 kinase, which phosphorylates lipids in the cell membrane, creating docking sites for proteins containing pH domains. This leads to activation of AKT, a kinase that promotes cell survival by inhibiting apoptosis pathways. Other RTKs recruit enzymes like phospholipase C-gamma, which cleaves membrane phospholipids to generate second messengers, small molecules like inositol trisphosphate and diacylglycerol that trigger calcium release and activate protein kinase C. The same receptor, activated by the same ligand, can trigger 5 or 6 different pathways at once, and the cell integrates all of them. The final response, whether to divide, differentiate, move, or die, depends on which combination of pathways is active, how strongly, and for how long. According to a 2024 study in PNAS Nexus on dynamic cell signaling, these pathways don't produce static responses. The intensity of signaling changes over time, shaped by feedback loops. Activated ERK, for example, can phosphorylate proteins that inhibit upstream steps in its own activation pathway, creating negative feedback that prevents over-signaling. Other pathways show positive feedback, where activation reinforces itself, creating bistable switches that lock cells into particular states. The timing matters too. Experiments with neuronal cells have shown that transient ERK activation, a brief pulse, can trigger proliferation, while sustained ERK activation for hours triggers differentiation into neurons. The pathway is the same. The outcome depends on duration. This temporal dimension adds enormous complexity to cellular decision-making. A cell doesn't just ask, is this pathway on or off? It asks, how strongly is it activated, and for how long, and in what temporal pattern? Pulses versus sustained activation. Oscillations versus steady states. The dynamics encode information, and cells integrate signals from multiple receptors simultaneously. An epithelial cell in a developing tissue might be receiving growth factor signals through one receptor, integrative signals through integrins that detect attachment to extracellular matrix, mechanical signals from tension in surrounding tissues, and metabolic signals about nutrient availability. Each signal activates its own pathways, but these pathways crosstalk extensively. Proteins activated by one pathway might inhibit or enhance another pathway. The cell essentially performs analog computation, summing and weighing these inputs to decide how to respond. Receptor tyrosine kinases aren't the only receptor type, but they illustrate the general principle. Receptors translate extracellular chemistry into intracellular biochemistry through conformational changes that trigger enzymatic activity. Other major receptor classes use variations on this theme. G-protein coupled receptors, the largest receptor family in humans with over 800 members, don't have intrinsic kinase activity. Instead, they activate G proteins, molecular switches similar to RAS that cycle between GDP-bound inactive and GTP-bound active states. Activated G proteins then trigger downstream enzymes that produce second messengers like cyclic AMP. Cytokine receptors lack kinase domains but recruit separate kinase proteins when activated. Ligand binding brings two receptors together, and cytoplasmic kinases called JAKs, Janus kinases, attach to the receptors and phosphorylate proteins called STATs, which dimerize and move directly into the nucleus to regulate gene expression. The diversity of receptor types and signaling mechanisms reflects evolutionary tinkering. Different receptor families arose at different points in evolutionary history, each optimized for particular types of signals and cellular contexts. But they all share the core logic. Binding changes shape, shape change triggers enzymatic activity. Enzymatic activity initiates a cascade that amplifies the signal and transmits it to the nucleus or other cellular compartments where responses are executed. What's striking is how much information is encoded not just in which receptor is activated, but in how it's activated. The concentration of ligand affects how many receptors are engaged. The spatial distribution of receptors on the membrane affects how efficiently they can dimerize. The presence of co-receptors or modifier proteins can tune receptor sensitivity, and once activated, the strength and duration of downstream signaling depends on regulatory mechanisms. Phosphatases that remove phosphates added by kinases, adapter proteins that compete for binding sites, feedback inhibitors that shut down pathways after they've been on for too long. Cells have evolved exquisite control over these systems because the consequences of error are severe. Too much signaling and cells divide when they shouldn't, leading to tumours. Too little, and tissues don't form properly, or don't repair damage, or can't mount immune responses. The balance is maintained through layers of regulation, fail-safes and checkpoints. But receptors only solve part of the problem. They tell individual cells what signals are present in their immediate environment. To build complex structures, organs, limbs, nervous systems, cells need more than binary on-off signals. They need positional information. They need to know not just that a signal exists, but how much of it there is, and how that amount compares to what their neighbours are experiencing. They need to read gradients, and doing that requires a different kind of cellular mathematics. Imagine you're standing on a beach at night. You can't see the ocean, but you can hear it. The sound of waves is louder when you face the water, quieter when you turn away. Without seeing anything, you know where the ocean is relative to your position. You've read a gradient, in this case, of sound intensity, and extracted positional information from it. Cells do something similar with chemical concentration. A morphogen gradient is a spatial pattern of molecular concentration, typically high near a source and decreasing with distance. The simplest way to create such a gradient is diffusion. A group of cells produces morphogen molecules, secreting them into the extracellular space. The molecules bounce randomly in Brownian motion, spreading outward. Simultaneously, cells throughout the tissue remove morphogen from the extracellular space, either by degrading it with extracellular enzymes or by internalizing it through receptor-mediated endocytosis. The result is a steady-state profile, production at the source, diffusion through space, removal by degradation and uptake. These three processes balance to create a concentration that decreases exponentially with distance from the source, typically spanning anywhere from tens to hundreds of cell diameters, depending on the morphogen's diffusion rate and degradation rate. According to research published in Development in 2025, the geometry of the tissue itself profoundly affects gradient shape. The extracellular space between cells isn't a smooth, open volume. It's a tortuous, porous medium, narrow gaps, dead ends, regions where diffusing molecules get temporarily trapped. Computer simulations that reconstruct the actual three-dimensional structure of zebrafish embryos from microscopy images show that morphogen molecules take longer, more winding paths through this complex geometry than simple diffusion models predict. And gradients are shaped by interactions with the extracellular matrix. Morphogen molecules don't just diffuse freely, they bind transiently to proteins embedded in the matrix, especially heparan sulfate proteoglycans, long chains of sugar molecules attached to core proteins. Research from 2024 using single-molecule imaging in living zebrafish embryos found that fibroblast growth factor 8, or FGF8, binds to these matrix molecules with different affinities at different locations. When bound, the morphogen diffuses more slowly. When released, it jumps quickly through the extracellular space before binding again. This creates what's called hindered diffusion. The morphogen alternates between a mobile state, diffusing rapidly through open spaces, and an immobile state bound to matrix components. The result is an effective diffusion rate slower than free diffusion in water, which spreads the gradient over a longer distance and makes it more stable. Cells measure this gradient by counting receptors. More precisely, they count activated receptors. At any given moment, morphogen molecules are colliding with cell surface receptors, binding, dissociating, rebinding. The rate at which receptors become occupied depends on the local morphogen concentration. High concentration means more frequent binding events, more receptors occupied at any given time. Low concentration means fewer occupied receptors, but cells don't just passively count, they actively shape what they're measuring. Receptor-mediated endocytosis, the process where ligand-receptor complexes are internalized into the cell, doesn't just relay the signal, it removes morphogen from the extracellular space. Every receptor that binds morphogen and gets internalized is removing that morphogen molecule from the pool available to neighboring cells. Cells act as sinks, degrading morphogen after internalizing it, preventing it from diffusing further. This means the gradient itself is shaped by cellular responses to it. High receptor expression in a particular region will steepen the gradient there, creating sharper boundaries between high and low morphogen zones. Conversely, if cells downregulate their receptors, the gradient flattens, extending further. The system is self-organizing. No central controller is positioning morphogen molecules or telling cells how many receptors to express. The gradient emerges from local interactions, production, diffusion, binding, degradation, playing out in parallel across thousands of cells. And here's what makes it powerful. Cells can use the same gradient to specify many different cell types by responding to different threshold concentrations. This is Wolpert's French flag model in action. In the developing spinal cord of vertebrates, a morphogen called Sonic Hedgehog, named improbably after the video game character, is secreted by cells along the floor plate, the bottom midline of the neural tube. Sh diffuses dorsally, creating a gradient that extends upward through the tissue. Cells measure the concentration of Sh they're experiencing and respond by activating different combinations of transcription factors. High Sh concentration activates one set of genes, medium concentration activates a different set, low concentration activates yet another set. And each combination of active transcription factors drives the cell toward a different fate, different neuron subtypes with different functions in the mature spinal cord. The thresholds are built into the gene regulatory networks. A gene might have an enhancer that requires a certain level of activated sh signaling to turn on. Another gene has a different enhancer with a different threshold. By stacking genes with different sensitivities to sh signaling, the cell effectively converts the analog gradient into discrete zones of gene expression. Research in Nature Communications from 2022, studying the mouse neural tube, found that the precision of these boundaries is remarkable, about 5% of a cell diameter. That's far more precise than you'd expect given that individual morphogen molecules diffuse randomly, that receptor activation is stochastic, that gene expression itself is noisy. How do cells achieve this precision? Part of the answer is time averaging. Cells don't make fate decisions based on a snapshot of morphogen concentration. They integrate signaling over hours, sometimes tens of hours, averaging out random fluctuations. A momentary spike in morphogen concentration doesn't trigger a fate change. Sustained signaling above threshold does. Another mechanism is mutual repression between transcription factors. Once a gene is turned on in response to a particular morphogen level, the proteins it produces often inhibit genes associated with other fates. This creates a form of cellular memory. Once a cell is committed to a particular expression state, it takes a much stronger signal to push it into a different state. The system is bistable, with distinct stable states separated by thresholds that are harder to cross than you'd expect from the noisy gradient alone, and cells use multiple gradients simultaneously. In the neural tube, Sh is opposed by bone morphogenetic proteins, or BMPs, secreted from the roof plate at the opposite end of the tissue. BMP signaling is high dorsally, low ventrally, the opposite pattern to Sh. Cells integrate both signals. essentially triangulating their position from 2 reference points rather than 1. This dual gradient system is more robust than a single gradient. If Shep production fluctuates, the position of a particular threshold concentration would shift. But if both SH and BMP fluctuate proportionally, perhaps due to changes in tissue size or temperature, the position where they meet particular combinations of concentrations remains relatively stable. The ratio of the 2 signals encodes position more reliably than either signal alone. Recent work has shown that gradients can scale with tissue size. If you grow an embryo that's 20% larger than normal, the morphogen gradient stretches proportionally, maintaining the same relative pattern of cell fates even though the absolute distances have changed. This suggests feedback mechanisms, possibly cells measuring not just absolute morphogen levels, but also the spatial derivative, the rate of change of concentration across neighbouring cells, adjusting their thresholds accordingly. The mathematics of gradient formation turn out to be surprisingly rich. In the simplest model, production, diffusion, degradation, the steady-state concentration profile is exponential decay, with a characteristic length scale determined by the square root of the ratio of diffusion coefficient to degradation rate. Increase diffusion or decrease degradation, and the gradient extends further. Decrease diffusion or increase degradation, and it shortens. But real gradients are more complex. Some morphogens exist in multiple forms, short-range forms that diffuse slowly and long-range forms that diffuse rapidly. Drosophila development uses a morphogen called decapentaplegic that exists both bound to extracellular matrix, remaining near where it's produced, and freely diffusing. The relative amounts of these forms tune gradient shape. Other morphogens are transported actively rather than passively. Wingless, another Drosophila morphogen, moves along specialized cellular protrusions called cytonemes, thin filopodia that extend from receiving cells towards source cells, creating direct delivery routes that bypass diffusion through extracellular space. This allows long-range signaling even for molecules that would otherwise diffuse poorly. And some gradients are read not by receptors on the cell surface, but by transcription factors inside cells. The bicoid protein in Drosophila embryos forms a gradient by diffusing through the cytoplasm of a syncytium, a common cytoplasm shared by thousands of nuclei before the embryo cellularizes. Bicoid is produced from mRNA localized at the anterior pole and diffuses toward the posterior, creating a concentration gradient that nuclei throughout the embryo can read directly without needing membrane receptors. In all these cases, the core principle is the same. Spatial variation in concentration is interpreted by cells as positional information. The gradient itself is a coordinate system, a chemical map that tells cells where they are and what they should become. But gradients alone can't explain all of multicellular coordination. Some cellular conversations need to be faster, more direct, more tightly coupled than diffusion allows. Some tissues need cells to act in perfect synchrony, heart muscle contracting as one, neurons firing in coordinated patterns, tissues maintaining electrical continuity. For this, evolution invented a more intimate form of connection, cells that physically link their interiors, sharing not just signals but their very cytoplasm, creating electrical and chemical unity across boundaries that would otherwise separate them. There's a different kind of conversation happening between cells, one that doesn't rely on molecules diffusing through extracellular space. It's faster, more intimate, more like cells merging their identities than maintaining separate boundaries. Gap junctions are microscopic tunnels connecting the cytoplasm of adjacent cells. Built from proteins called connexins, these channels don't just transmit chemical signals, they physically link the interiors of neighbouring cells, allowing ions, small molecules, and electrical currents to flow directly from one cell to another. Each channel is composed of 12 connexin proteins, 6 from each cell. The 6 connexins from one cell assemble into a hexameric structure called a connexin or hemichannel. This hemichannel inserts into the cell membrane and extends across it. When 2 cells come close together, their hemichannels dock end-to-end across the narrow gap between their membranes, forming a complete channel that spans both cells. The result is a pore about 1.5 nanometers wide, large enough for ions and small molecules up to roughly 1,000 Daltons to pass through, but small enough to prevent proteins and other large molecules from crossing. Through these pores, sodium ions, potassium ions, calcium ions, and metabolites like glucose, ATP and various second messengers can move directly from cell to cell. In tissues with gap junctions, cells aren't truly separate. They form electrical and metabolic networks, syncytia in which boundaries still exist but don't prevent direct exchange. The most dramatic example is the heart. Your heart contains more than 1 billion cardiac muscle cells. When one cell depolarizes, when sodium ions rush in through voltage-gated channels, flipping the membrane potential from negative to positive, that electrical change must propagate to its neighbors within milliseconds. The wave of depolarization sweeps through the ventricles, triggering contraction that pumps blood to your body. This requires exquisitely precise coordination. If different regions of the heart contracted out of sync, the pumping action would fail. Blood would pool. Within minutes, you'd lose consciousness. Within minutes more, brain death would begin. Gap junctions make cardiac conduction possible. According to research on cardiac gap junction function, connexin 43, the primary gap junction protein in ventricular heart muscle, forms dense clusters at the ends of cardiac myocytes, where cells meet at structures called intercalated discs. These clusters contain hundreds to thousands of gap junction channels packed tightly together. When a cardiac myocyte fires an action potential, positive current flows into the cell. Some of that current exits through gap junction channels into neighboring cells. The incoming current depolarizes those neighbors toward their threshold, triggering their action potentials, which generate more current that flows to the next cells, propagating the wave. The speed of this propagation depends on how many gap junction channels connect the cells. More channels mean lower electrical resistance between cells, faster current flow, faster propagation. Along the long axis of cardiac muscle fibers, where gap junctions are abundant, the action potential travels at about 40 centimeters per second. Perpendicular to the fibers, where gap junctions are less dense, it travels at about 20 centimeters per second. The heart is anisotropic. Its electrical properties depend on direction, because gap junctions are distributed asymmetrically, and the system is dynamic. Connexins have remarkably short half-lives in the plasma membrane, between 1 and 5 hours. The gap junction channels you have in your heart right now weren't there yesterday. They're constantly being synthesized, transported to the membrane, assembled into junctions, internalized and degraded, replaced by new channels in a continuous cycle. This rapid turnover allows the heart to adjust its coupling in response to stress, injury or disease. During a heart attack, when blood flow to part of the cardiac muscle is blocked, connexin 43 redistributes within minutes to hours. Gap junctions that were concentrated at the cell end start appearing on the sides of cells, a phenomenon called lateralization. The electrical coupling pattern changes, conduction becomes irregular, and this creates substrate for arrhythmias. Research on gap junction remodeling in diseased hearts has found that chronic heart failure, cardiomyopathy, and other cardiac diseases all show similar changes. Reduced connexin 43 expression, altered gap junction distribution, decreased cell-to-cell coupling. These changes correlate with increased risk of sudden cardiac death from ventricular arrhythmias. But here's something unexpected. Hearts can function with far fewer gap junctions than you'd think. Mice genetically engineered to express only 5% of normal connexin 43 levels still develop normally and have surprisingly normal heart function. Their cardiac conduction is slowed, but not catastrophically. This has led researchers to propose that gap junctions might not be the only mechanism of electrical coupling between heart cells. Alternative theories suggest ephaptic coupling, electrical communication through electric fields in the narrow extracellular space between cells without direct cytoplasmic connection. When an action potential travels along one cell, it generates electric fields that can influence neighboring cells if they're close enough together. Some computational models suggest that ephaptic coupling might account for a significant fraction of cardiac conduction, especially in regions where cells are tightly packed. The truth likely involves both mechanisms, gap junctions and ephaptic coupling working together in what's called mixed-mode conduction. The gap junctions might serve not just as electrical conduits, but as structural elements that keep cell membranes close enough together for ephaptic effects to matter. Gap junctions aren't unique to the heart. They're found throughout the body in tissues that require coordinated activity. In smooth muscle, the involuntary muscle in blood vessel walls, intestines, the uterus, gap junctions couple cells into functional units. When smooth muscle contracts to narrow a blood vessel or push food through your intestines, the contraction spreads through gap junction-mediated electrical coupling. The uterus during childbirth dramatically upregulates connexin 43 expression, transforming from a quiescent organ into a coordinated contractile machine. In the nervous system, gap junctions form electrical synapses between neurons. Unlike chemical synapses, where neurotransmitter molecules cross a synaptic cleft, electrical synapses transmit signals directly as current flow through gap junction channels. These synapses are bidirectional, faster than chemical synapses, and allow for extremely precise synchronization of neuronal firing. Retinal neurons are extensively coupled by gap junctions, creating networks where light signals detected by photoreceptors spread laterally through the retina, enhancing contrast and sensitivity. Inhibitory interneurons in many brain regions use gap junctions to synchronize their firing, generating rhythmic activity patterns that coordinate larger neural networks. And gap junctions couple non-neuronal cells. Astrocytes, star-shaped glial cells that support neurons, form vast networks connected by gap junctions. Calcium waves propagate through these astrocytic networks, coordinating metabolic support across large regions of the brain. Some evidence suggests astrocyte networks modulate neural activity, though the mechanisms and functional significance remain debated. Even in development, gap junctions play coordination roles. During the earlier stages of embryogenesis, when the embryo is just a handful of cells, gap junction coupling begins appearing. In mouse embryos, connexin 43 starts assembling into gap junctions during compaction at the 8-cell stage, precisely when cells need to start making collective decisions about which will form the inner cell mass and which will form the trophoblast. Experiments blocking gap junction formation during this critical period show that embryos can still develop, suggesting gap junctions aren't absolutely required for early mammalian development, but the process is less reliable, more prone to errors. Gap junctions might provide a communication backup, a way for cells to share metabolic resources and coordinate responses to stress, even when other signaling mechanisms are still being established. The molecular diversity of connexins allows functional specialization. Humans have 21 different connexin genes, expressed in different combinations in different tissues. Different connexins form channels with different properties, different conductances, different permeabilities to specific molecules, different regulatory mechanisms. Connexin 40 and connexin 43, both expressed in some cardiac tissues, form channels with different gating properties, different sensitivities to voltage, pH, and phosphorylation. Heteromeric channels, those containing multiple connexin types in the same connexin, can have intermediate properties, creating a spectrum of coupling strengths and selectivities. This means tissues can tune their gap junction networks precisely, adjusting not just how many channels they have, but what kinds, creating communication networks with specific electrical and metabolic properties optimized for particular functions. And gap junctions can be gated, opened and closed in response to signals. Connexin channels close in response to low pH, high calcium concentrations, and certain phosphorylation events. This allows cells to isolate themselves from neighbors when stressed or damaged, preventing the spread of toxic molecules or metabolic disturbances through the network. During a heart attack, for example, dying cells become acidic and leak calcium. Their gap junction channels close, electrically isolating the damaged region and limiting how far the injury spreads. It's a form of tissue-level damage control, sacrificing coupling to protect the larger network. But gap junctions are only part of the cellular communication infrastructure. They're fast and direct, but limited to neighboring cells and small molecules. To coordinate over longer distances, to integrate signals from multiple sources, to remember what signals have been received even after those signals fade, cells need more sophisticated machinery. They need gene regulatory networks that can store information, make decisions, and maintain cellular identity through time. They need systems that can translate transient chemical signals into permanent changes in what a cell is and what it does. A signal arrives at a cell, a receptor binds it. A cascade activates. Transcription factors enter the nucleus and turn genes on. Then the signal disappears. The ligand dissociates from the receptor. The cascade shuts down. The transcription factors get degraded or exported from the nucleus. And yet, the cell remembers. Hours, days, sometimes weeks after the signal has vanished, the cell still expresses the genes that were activated. It has changed its identity, permanently, based on a transient chemical instruction. This is cellular memory, and it happens without anything like a brain, without neurons or synapses or any specialized memory organ. The mechanism is gene regulatory networks, circuits of genes that regulate each other through transcription factors, creating stable patterns of gene expression that persist long after the initiating signal is gone. Here's how it works. A transcription factor is a protein that binds to specific DNA sequences near genes, promoters and enhancers, and either activates or represses their transcription. When a gene is transcribed, its DNA is copied into messenger RNA, which is then translated into protein. Some of those proteins are transcription factors themselves, which can then regulate other genes, including sometimes the very genes that encode them. This creates feedback loops. A gene whose protein product activates its own transcription is called an auto-regulatory positive feedback loop. Once the gene turns on and starts producing protein, that protein reinforces its own expression, keeping the gene on even after the signal that initially activated it has disappeared. Conversely, genes can inhibit their own expression, negative feedback, creating oscillators or homeostatic controls that prevent overexpression, and genes regulate each other in complex cross-regulatory networks. Gene A activates gene B inhibits gene C inhibits gene A. These circular patterns of activation and inhibition can create bistable switches, systems with two stable states that the cell can flip between but doesn't easily occupy intermediate positions. According to research published in eLife in 2024 on cell fate determination, these network architectures function like computational logic gates. Some transcription factors work as AND gates. Both input A and input B must be present for the output gene to turn on. Others work as OR gates. Either input A or input B is sufficient. The specific logic of how transcription factors combine to regulate target genes determines how the network behaves, how it responds to signals, and what stable states it can achieve. Think of it as a landscape. Each possible combination of gene expression states, gene A on, gene B off, gene C on, and so forth, is a point in a high-dimensional space. Some points in this space are valleys, stable attractors where the network tends to settle. Others are hilltops, unstable states the network quickly leaves. The topology of this landscape, where the valleys are, how deep they are, what ridges separate them, determines what cell types the network can produce. A stem cell occupies a valley in this landscape. When it receives a differentiation signal, that signal effectively tilts the landscape, making the stem cell valley shallower and other valleys deeper. The cell rolls downhill into a new valley, a differentiated cell type, and once there, it's trapped. The network stabilizes in a new configuration that maintains itself through feedback loops. This is why differentiation is usually irreversible. Once a cell is committed to being a neuron or a muscle cell or a skin cell, the gene regulatory network locks into that state. Genes characteristic of the neuron fate activate each other and repress genes for alternative fates. The system is bistable, neuron valley or muscle valley, not halfway between. Reprogramming, forcing a differentiated cell back to pluripotency or converting it to a different cell type, requires overriding these stable states. The discovery that this is possible at all, that you can take an adult skin cell and reprogram it into a pluripotent stem cell by forcing expression of just 4 transcription factors, won Shinya Yamanaka the Nobel Prize in 2012. He essentially forced cells out of their valley and into a different one by overwhelming the endogenous regulatory network with exogenous factors. But even in normal development, cells navigate this landscape through carefully orchestrated changes in gene expression, driven by morphogene signals, cell-cell contacts, and internal timers. In the developing neural tube, cells receiving high sonic hedgehog signaling activate transcription factors like NKEX 2.2 and FOXA2, which promote ventral neuron fates. These transcription factors inhibit dorsal transcription factors like PAX6 and PAX7. Meanwhile, cells far from the sheesh source, receiving little or no signal, activate PAX6 and PAX7. which inhibit the ventral factors. Mutual repression creates sharp boundaries. Cells are either ventral or dorsal, not intermediate. Within these broad domains, finer distinctions emerge through combinatorial transcription factor codes. A cell expressing NKES 2.2 but not OLIG2 becomes one neuron subtype. A cell expressing both becomes another. The specific combination of active transcription factors, the cell's position in gene expression space, determines its fate, and once established, these states are reinforced through chromatin modifications. DNA in eukaryotic cells is wrapped around histone proteins, forming structures called nucleosomes. The accessibility of DNA to transcription factors depends on how tightly it's packaged. Regions of open chromatin, where nucleosomes are loosely packed or modified with activating chemical marks, are accessible and actively transcribed. Regions of closed chromatin, where nucleosomes are tightly packed or marked with repressive modifications, are inaccessible and silenced. When a transcription factor activates a gene, it often recruits chromatin remodeling complexes that open the chromatin at that locus, making it easier for the gene to stay active. When a gene is repressed, repressive complexes compact the chromatin, making it harder to reactivate even if the repressor later disappears. The chromatin state itself becomes a form of memory, a physical record of which genes have been active and which have been silent. This is epigenetic memory, heritable changes in gene expression that don't involve changes to the DNA sequence itself. When a cell divides, both daughter cells inherit not just the DNA but also the chromatin modifications and transcription factor networks that determine which genes are accessible and which are silenced. This is how a differentiated cell produces two differentiated daughters, how memory persists through cell division. The stability of these states is remarkable. A neuron in your brain has been a neuron for years, decades, maintaining its identity through countless rounds of protein turnover, constantly expressing neuron-specific genes and repressing everything else. The gene regulatory network that defines neuronness has been active continuously, self-reinforcing through feedback loops that survived every meal you ate, every night you slept, every stress your body experienced. But gene regulatory networks aren't just about memory, they're computational devices that integrate multiple inputs and make decisions. Research from 2024 on neural network models of cell fate shows that gene regulatory networks function as attractor-based systems, capable of responding to multiple instructive signals. Depending on the timing and identity of signals, the same network can drive cells toward different fates. Early exposure to signal X pushes cells into valley A. Later exposure to signal Y pushes them into valley B. The network computes an output, cell fate, based on the sequence and combination of inputs it receives. This allows developmental systems to generate diverse cell types from the same genetic information. Every cell in your body has the same genome, but vastly different gene expression profiles. A liver cell and a neuron are running different programs specified by different stable states in the same underlying gene regulatory network, and networks can be hierarchical. Master regulatory transcription factors at the top of the hierarchy control many downstream genes. Changing a master regulator can flip an entire network from one stable state to another. This is why Yamanaka's four factors, OST4, SOX2, KLF4, and C-MYC, can reprogram cells. They're master regulators that, when overexpressed, reconfigure the entire network. The logic of how transcription factors interact matters profoundly for network behavior. Research on gene regulatory logic has shown that ND gate regulation, where multiple transcription factors must bind cooperatively to activate a gene, creates different dynamics than OR gate regulation, where any one of several factors is sufficient. AND gates require coincidence detection, making them useful for integrating multiple signals. OR gates provide redundancy, making networks more robust to loss of any single factor. Different regulatory logic also affects noise sensitivity. Gene expression is inherently stochastic. Transcription and translation occur in random bursts, creating cell-to-cell variability even in genetically identical populations. Some network architectures buffer this noise, using negative feedback to maintain consistent expression levels. Others amplify noise, using it as a source of diversity that allows different cells to explore different fates, even from identical starting conditions. During development, both strategies are used. Early embryonic cells often show high gene expression noise, creating heterogeneity that seeds fate diversity. As development progresses and cells commit to specific lineages, noise is suppressed by feedback mechanisms that lock in stable states. The interplay between deterministic signaling, morphogen gradients providing positional information, and stochastic fluctuations, noise creating variability, shapes how development unfolds. Signals bias probabilities, but don't absolutely determine fates. A cell receiving intermediate sheesh signaling might flip a genetic coin, becoming one fate or another based partly on random fluctuations amplified by the gene regulatory network. This stochasticity isn't a bug. It's a feature that creates diversity, resilience, and adaptability. Even in adult tissues, some degree of noise persists, allowing tissues to respond flexibly to damage or changing conditions. What's extraordinary is that all of this, memory, computation, decision-making, information storage, happens through nothing more than proteins binding to DNA, regulating transcription, creating feedback loops that stabilize specific patterns of gene expression. There's no homunculus directing the process, no central controller deciding which genes should be on or off. The network itself is the intelligence, the memory, the decision maker. And when these networks malfunction, the consequences are severe. Cancer is a disease of cellular communication gone wrong. Not entirely, not even primarily. Cancer involves mutations in DNA, changes to the genetic code that alter how proteins function. But many of the most important cancer-causing mutations affect genes that encode components of signaling pathways: receptors, kinases, transcription factors, the molecular machinery that cells use to listen to their neighbours and respond to their environment. When these signaling systems malfunction, cells stop obeying the rules. They divide when they shouldn't. They ignore signals telling them to stop. They migrate to places they don't belong. They resist signals that should trigger their death. The conversation that coordinates multicellular life breaks down, and individual cells revert to a more primitive mode. Growth for growth's sake. Competition rather than cooperation. Consider RAS. This small protein, mentioned earlier as part of the MAP kinase cascade, acts as a molecular switch in growth factor signaling. When a receptor tyrosine kinase activates, it recruits proteins that cause RAS to exchange GDP for GTP, turning it on. Active RAS then triggers the kinase cascade that ultimately activates ERK and drives cell division. But RAS has intrinsic GTPase activity. It hydrolyzes GTP back to GDP, turning itself off after a brief period. This ensures signaling is transient, pulsed, controlled. According to research published in 2024 on oncogenes, mutations in RAS genes are found in roughly 25% of all human cancers. These aren't random mutations, they're highly specific, occurring at particular positions in the protein, most commonly at codons 12, 13, and 61. These mutations do something precise. They impair RAS's ability to hydrolyze GTP. Mutant RAS stays locked in the active GTP-bound state. It signals continuously, regardless of whether growth factors are present, regardless of what signals the cell is receiving from its environment. The switch is jammed on. The result is uncontrolled activation of downstream pathways. ERK remains active, constantly driving transcription of genes that promote cell division. The cell proliferates when it should be quiescent. It ignores contact inhibition, the mechanism that normally stops cells from dividing when they're crowded. It becomes insensitive to growth suppressive signals. RAS mutations are particularly common in pancreatic cancer, where they appear in more than 90% of tumours and in lung and colorectal cancers. For decades, RAS was considered undruggable. Its structure lacks obvious binding pockets for small molecule inhibitors, but recent advances have produced drugs that specifically target the KRAS G12C mutation, one of the common variants, by binding to the mutant protein and locking it in an inactive state. These drugs work. Lung cancers with KRAS G12C mutations shrink when treated with these inhibitors, but the effect is temporary. Within months, most tumours develop resistance. cancer cells find alternative signaling routes, activating parallel pathways that bypass the blocked RAS protein. The network is redundant enough that shutting down one node isn't sufficient. Other common cancer mutations affect receptor tyrosine kinases directly. GR2, a receptor in the EGFR family, is amplified, present in many extra copies, in about 20% of breast cancers. More HER2 protein means more receptors on the cell surface, more signaling even with normal levels of ligand. The cell behaves as if it's constantly receiving growth signals. Or consider mutations that create constitutively active receptors, proteins that signal even without ligand binding. Some EGFR mutations found in lung cancer cause the receptor to dimerize and activate spontaneously. The cell no longer needs external growth factors. It provides its own signals decoupled from environmental cues. The PI3K AKT pathway, another major signaling network downstream of RTKs, is dysregulated in a vast number of cancers. PIK3CA, the gene encoding one of the PI3K catalytic subunits, is one of the most frequently mutated genes in cancer, altered in many breast, colorectal, and endometrial tumours. Activating mutations in PIK3CA increase the enzyme's activity, driving excessive AKT signaling that promotes cell survival and growth, or PTEN is lost. PTEN is a phosphatase that opposes PI3K. It removes the phosphate groups that PI3K adds, shutting down the pathway. When PTEN is deleted or inactivated by mutation, PI3K signaling runs unopposed, AKT stays active, cells resist apoptosis, survive in conditions that should kill them, accumulate further mutations because they're not being eliminated by quality control mechanisms. Analysis from the Cancer Genome Atlas, which profiled genetic alterations in more than 9,000 tumours across 33 cancer types, found that 89% of tumours have at least one driver mutation in 10 major signalling pathways. 89%. Nearly every cancer involves corrupted communication. Some tumours have mutations in multiple pathways. Pancreatic cancers often have mutant CRAS plus inactivated 53 plus lost SMAD4. 3 critical signalling nodes simultaneously compromised. The redundancy and crosstalk in signalling networks means that disrupting one pathway isn't enough. Cells find ways around it. Multiple hits are required to fully transform a cell from normal to malignant, and tumour suppressor genes, genes whose normal function is to restrain growth, are frequently lost. P53, the guardian of the genome, is mutated or deleted in over half of all human cancers. P53 normally responds to DNA damage and cellular stress by halting cell division, and either repairing the damage or triggering apoptosis. Without functional P53, cells with damaged DNA continue dividing, accumulating more, mutations progressing toward malignancy. Retinoblastoma protein, PRB, normally prevents cells from entering this phase of the cell cycle until appropriate growth signals are received. When PRB is lost, cells divide regardless of external signals. The brake is removed. What's striking is that many cancer-causing mutations don't create entirely new functions. They're often subtle changes, point mutations, small deletions, gene amplifications, that tip the balance of existing signaling systems. A protein that normally operates 10% of the time now operates 50% of the time. A feedback inhibitor that normally shuts down after an hour now shuts down after 10 hours, or not at all. The systems are robust by design, buffering against small fluctuations. But cancer mutations exploit specific vulnerabilities, targeting the control points, the negative regulators, the checkpoints that normally prevent runaway signaling. And once signaling is dysregulated, it creates conditions for further dysregulation. A cell dividing too frequently experiences more DNA replication errors. A cell surviving past its normal lifespan accumulates more mutations. Defective signaling snowballs into genomic instability, which produces more defective signaling. Developmental defects offer another window into what happens when cell communication fails. These aren't cancer, cells aren't proliferating out of control, but the consequences can be equally severe. Holoprosencephaly, a severe birth defect where the forebrain fails to properly divide into 2 hemispheres, is often caused by mutations in sonic hedgehog signaling. Without proper signaling, midline structures in the developing face and brain don't form correctly. The result ranges from mild facial abnormalities to profound malformations incompatible with survival. charge syndrome, a constellation of birth defects affecting the heart, eyes, ears, and other organs, results from mutations in CHD-CERN, a chromatin remodeling protein involved in gene regulation downstream of multiple signaling pathways. When gene regulatory networks can't respond properly to developmental signals, coordinated tissue formation fails. Even subtler signaling defects can have lifelong consequences. Mutations in Notch signaling, a pathway critical for specifying cell fates during development, cause Alagille syndrome, characterized by liver, heart, and skeletal abnormalities. The signaling doesn't fail completely, but it's impaired enough that tissue architecture forms abnormally. What all these examples reveal is how dependent multicellular life is on precise, reliable cellular communication. Development requires cells to coordinate across vast scales, millions of cells making synchronized decisions about what to become and where to go. Tissue maintenance requires constant signaling to regulate cell division, death, and differentiation. Immune responses require recognition and coordination across cell types and tissues. When communication fails, through mutation, through developmental error, through infection or injury, the consequences cascade. A cell that can't hear its neighbours loses context. A tissue that can't coordinate its cells loses structure. An organism whose cells don't cooperate loses coherence. But when it works, the result is almost miraculous. You started as a conversation. One cell, a fertilized egg, began dividing. 2 cells, then 4, then 8. At first, they were identical, totipotent, capable of becoming anything. But then, signals began flowing between them. Gap junctions formed. Cells started secreting morphagens. Receptors activated. Cascades fired. Gene regulatory networks clicked into stable states. And from chemistry alone, from molecules diffusing and binding and phosphorylating other molecules, form emerged. Groups of cells that were receiving high concentrations of certain signals differentiated one way. Groups receiving low concentrations differentiated another way. Cells at boundaries between these regions adopted intermediate fates. The embryo, originally a uniform ball of cells, began to regionalize, anterior and posterior, dorsal and ventral, left and right. Some cells started producing signals that told neighbouring cells to move. Those cells extended protrusions, detached from their neighbours, migrated along chemical gradients to new locations. There they received different signals, activated different gene programs, became different cell types. Tissues folded, forming sheets and tubes. The neural tube zipped shut along the back of the embryo, cells on either side meeting precisely at the midline through a choreography coordinated by morphogen gradients and cell adhesion molecules. The gut tube formed through similar folding, creating an interior space from an external surface, cell death sculpted form. In your developing hands, cells between your finger primordia received signals triggering apoptosis, programmed cell death. They died on schedule, carving gaps that became the spaces between your fingers. Without that coordinated death, you'd have webbed hands like a duck or a frog. All of this transformation from one cell to trillions, from symmetry to asymmetry, from potential to differentiated form, happened through cellular communication. Through signals sent, received, integrated, remembered. By the time you were born, you contained roughly 26 billion cells, organized into more than 200 cell types, arranged in precise anatomical structures. Your heart had formed 4 chambers, valves between them opening in one direction. Your lungs had branched into airways ending in alveoli. Your nervous system had wired itself, neurons sending axons to specific targets, guided by gradients of attractive and repulsive cues, forming synapses in patterns that encode reflexes and later, after birth, learned behaviors. Every cell in your body knew what it was and where it belonged because of conversations, chemical, dialogues that specified identity, coordinated development, built complexity from simplicity. And those conversations didn't stop at birth. They continue, constantly maintaining what was built. Your skin sheds dead cells from its outer surface every day, millions of them, and replaces them from stem cells in the basal layer. Those stem cells divide and their daughters migrate outward, differentiating through stages as they move, receiving signals from the extracellular matrix and neighbouring cells that tell them when to stop dividing and start producing keratins that toughen the skin. Your intestinal lining turns over every few days, the fastest renewing tissue in your body. Stem cells at the base of intestinal crypts receive WUNT signals that keep them proliferative. As their descendants move up and out of the crypt, WUNT signaling decreases, BMP signaling increases, and the cells differentiate into absorptive enterocytes, or mucus-secreting goblet cells, or hormone-producing enteroendocrine cells, different fates specified by different combinations of signals. Your immune system patrols constantly, white blood cells circulating through blood and lymph, sampling the molecular landscape, looking for foreign antigens or stressed cells that need to be eliminated. When they find a threat, they communicate, releasing cytokines that recruit other immune cells, presenting antigens to T cells to coordinate adaptive responses, forming immunological synapses where cells exchange information through direct contact. When you cut yourself, the wound healing response involves extensive cellular communication. Platelets at the wound site release growth factors and clotting factors. Immune cells infiltrate, attracted by chemical signals, clearing debris and fighting infection. Fibroblasts migrate in, receiving signals that tell them to proliferate and deposit extracellular matrix. Epithelial cells at the wound edge receive EGF and TGF-beta signals that trigger them to migrate and cover the wound. Blood vessels sprout new branches guided by VEGF gradients, re-establishing circulation. The entire process, clotting, inflammation, tissue regeneration, remodeling, is a coordinated multicellular response, orchestrated entirely through cell signaling. Even your thoughts depend on cellular communication, though of a more specialized kind. Neurons communicate through synapses, junctions where electrical signals in one cell trigger the release of neurotransmitters that bind receptors on another cell, generating new electrical signals. Each thought, each sensation, each movement is a pattern of activity flowing through networks of neurons, the pattern itself constituted by millions of conversations between cells. And the brain's ability to learn, to form memories, to change based on experience depends on modulation of those cellular conversations. Long-term potentiation, the strengthening of synapses with repeated activation, involves signaling cascades that change gene expression in the postsynaptic neuron, increasing the number of receptors, enlarging the synapse, making future signals more effective. Memory is physically encoded in the connectivity and strength of cellular communication channels. All of this is happening right now as you read this. Trillions of signals per second flowing between cells in your body. Morphagen gradients maintaining tissue boundaries. Gap junctions coupling cells into functional networks. Receptor tyrosine kinases responding to growth factors, integrating multiple inputs through crosstalk between pathways. Gene regulatory networks maintaining cell identities established during development, or adjusting those identities in response to new signals. The conversation that built you continues to sustain you. And it's not just internal. Your cells communicate with the external world, with bacteria in your gut, with the immune system's constant surveillance of the cellular landscape, with sensory receptors detecting light and sound and chemical stimuli and translating them into signals your nervous system can process. The boundary between self and environment is porous, mediated by receptors on cell surfaces constantly sampling, interpreting, responding to molecular information from outside. What's remarkable is how reliable it all is. Development could fail in countless ways, signals could be misinterpreted, gradients could be noisy, cells could commit to the wrong fates. But robust mechanisms, feedback loops, redundant pathways, error correction through cell competition and apoptosis, buffer against failure. The system is resilient because it's been refined by 4 billion years of evolution, every generation testing variants, selection keeping what works. Yet it's not perfect. Mutations accumulate. Communication systems degrade with age. Cancer exploits vulnerabilities. Developmental defects remind us that the process is probabilistic, not deterministic, that there are failure modes built into the complexity. But when you step back and consider the scope of what's being accomplished, transforming a single cell into an organism of trillions, maintaining that organism for decades, coordinating responses to an unpredictable environment, all through nothing more than molecules binding to each other and changing shape, it's staggering. There's no blueprint being read, no central controller issuing commands, No mind orchestrating the process, just cells talking to each other continuously in a chemical language refined over evolutionary time. Before there was language, there was chemistry. Before there was thought, there was signal transduction. Before there was you, there were 37 trillion decisions happening simultaneously, negotiating form and function through molecular conversation. The molecular ballet continues, every heartbeat coordinated by gap junctions, every wound healed by growth factor cascades, every memory encoded by synaptic plasticity, every immune response mounted through cytokine signaling. We began by asking how cells communicate to build complex organisms. The answer is, through every mechanism evolution could devise, diffusible signals creating gradients, direct connections through gap junctions, receptor-mediated signal transduction triggering intracellular cascades, gene regulatory networks storing information and making decisions, feedback loops creating stability and switches, crosstalk between pathways integrating multiple inputs. Each mechanism solves particular problems. Morphagen gradients provide positional information across distances. Gap junctions provide rapid, synchronized coordination. Receptor cascades amplify weak signals and allow integration of multiple cues. Gene networks create memory and maintain identity through time. Together they constitute the cellular communication infrastructure that makes multicellular life possible. And understanding these systems, how signals are sent, received, transduced, integrated, remembered, is understanding what life is at the most fundamental level. Not genes alone, not proteins alone, not even cells alone, but the relationships between cells, the information flowing between them, the coordination that emerges from molecular conversation. Life, at this scale, is communication. It's the difference between a collection of isolated cells and a coherent organism. It's what transforms chemistry into biology, molecules into meaning, chaos into the ordered complexity of a body that knows itself, maintains itself, heals itself. The conversation that began 800 million years ago when cells first stayed together after division continues in every living thing, in you, right now, in the signals flowing between your cells, maintaining the pattern that is you. Before there was language, there was this. And it's still speaking, if you know how to listen.